It is my pleasure today to introduce Dr. Rayna Bell, who is coming to us today from the Smithsonian Institution, where she's a curator of reptiles and amphibians at the National Museum of Natural History. So Rayna got her undergraduate degree at UC Berkeley, and then she went on and did a PhD with Kelly Zamudio at Cornell. After that, she went back to Berkeley and got a Chancellor's postdoctoral fellowship. And after that, she ended up at the Smithsonian. So I'm really excited today to hear about her work on phenotypic diversification in frogs in Africa. And I'm supposed to remind you to, if you want to stay and ask some questions and eat some food afterwards, please, you are welcome to do that. So without further ado, I'll turn it over to Rayna. Thanks.
Okay, great. Thanks, everybody. Is this loud enough? I think I can make it. Okay. How's that? Okay, great. Thanks, everyone, for sticking around this afternoon. So island biodiversity has inspired evolutionary biologists for centuries. And as a graduate student, I became particularly interested in how amphibians have been able to colonize oceanic islands in some contexts and how this has happened, because it's sort of this exception to the rule of things that you're supposed to find on oceanic islands. And so today I'll be talking about diversification of island amphibians in 3 different contexts. So I'll be talking about the early stages of population divergence on a land-bridge island, and overseas dispersal to colonize oceanic islands, and then finally in situ diversification within an island archipelago. So there are 2 main ways that you can get new species on islands. And so you can either accumulate new species when you have a dispersal event, either from the continent or adjacent islands, and those dispersers eventually differentiate from those sources and become endemic to their new home. Or you can have in situ diversification of island residents. And the relative importance of these 2 processes, so dispersal versus in situ diversification, can vary with a number of just characteristics of an archipelago. And so with increasing island age, size, and remoteness, in situ diversification predominates, versus in relatively young archipelagos that are large and are near continental sources, where dispersal is a dominant process leading to the accumulation of new species. So those processes hold for oceanic islands, where dispersal to those islands is a critical step to getting species in the first place. But the processes are pretty different on continental or land-bridge islands. And in these island systems, we often end up with populations arising on islands due to vicariance, when the sea levels rise and those parts of the continental shelf are now shallow seas, and what were once mountains on the edge of the continental shelf become islands. And so over the last several million years, we've had these global glacial cycles all around the world that have led to these rising and retreating sea levels. And these are resulting in these cyclical patterns of isolation and connectivity for the populations that inhabit these parts of the continental shelf. And so some of the most recent fluctuations have been in the last 15,000 years, but again, these cycles have been happening for millions of years. And so these types of island systems present a nice contrast to oceanic islands, which tend to be on much older geological time periods, whereas again, these processes on land-bridge islands tend to be more recent. And so even within these more recent periods of isolation and connectivity, we have organisms differing in their responses to these processes that are happening around them. So we can imagine that species that are good dispersers across saltwater barriers aren't necessarily going to experience periods of isolation, or at least not as strongly as other organisms, whereas organisms that are poor dispersers across saltwater barriers might not have any gene flow during periods of marine incursion, and they might start to accumulate both genetic and phenotypic differences that make it such that even when the sea levels retreat and they come back into contact, or potentially back into contact with the rest of the mainland population, they are no longer interbreeding, or at least not as freely. So for the last 10 years or so, I've been working in the Gulf of Guinea archipelago. And this archipelago is comprised of 1 land-bridge island and 3 oceanic islands. And they're situated along the Cameroon volcanic line, which is not a hot spot, so I'll just clarify that right now. But it extends into the Gulf of Guinea, and then it sort of makes a Y shape into Cameroon and forms the Cameroonian highlands, which some consider to be sky islands. So the first island in the chain is Bioko Island, and the volcanoes that comprise this island are about 3 million years old, and it's part of the continental shelf. It's presently isolated from the rest of the continent by a shallow sea, and over the last 3 million years of its history, that shallow sea has risen and retreated multiple times, and so presumably that fauna has been isolated and connected, or potentially isolated and connected multiple times. And that is really in contrast to the rest of the islands in the archipelago. So the rest of the islands are volcanic. They arose from the seafloor. They've never been connected to the rest of the continent. The first of these islands is Principe, which is about 30 million years old. And it's presently about 1/4 of the size that it was at its peak. And that's because it's so old that large parts of it have eroded off. The next island is São Tomé. It's still quite large, and it's about 13 million years old. And then the last island is Annobón, which is really, really tiny. I don't know if you can see it that well on this slide, but on the part of the island closest to you, that's the runway. And it basically takes the whole island to land a plane there. So again, all of these islands arose as volcanoes from the ocean floor, and so everything that is on these islands had to disperse overseas to get there. And so as a early PhD student, what I was really inspired about with this system was that I would be able to look at these different processes of dispersal and in situ diversification in these very different geological contexts. So in islands that vary in age and distance from the continent, and then also ones that had this history of connectivity and ones that didn't. And what's really neat about this is you can tell just by looking at the patterns of biodiversity and endemism on the islands that these differences in geological history have been important in shaping patterns of diversity and endemism. So Bioko Island, which is the land-bridge island, has really high species diversity for an island of its size, but almost all of this diversity is shared with the adjacent continent, so shared with Cameroon and Gabon and Central African rainforests. And a few species are considered endemic, but for the most part, everything there is maybe a slightly distinct subpopulation of the continent, but not very genetically distinct. And so this could either indicate that there's been migration throughout the history of this island, or perhaps that the selective environments between the island and the rest of the continent are pretty similar. And now when we shift to the oceanic islands, we see that there's not as much diversity, but almost all of that diversity is endemic to one or perhaps a couple islands in the archipelago. And so even though these islands are isolated and everything has had to disperse to get there, because they've been sitting out in the ocean for a really long time, they have accumulated a lot of species, and there's been time for divergence and endemism to build over time. One of the things that's a little bit surprising about these islands is they have a lot of species that come from groups that you don't expect to find on oceanic islands. So there are endemic shrews, which if you don't know that much about shrews, they have very high metabolic rates. They have to eat every couple hours. And I don't think they're very good at swimming in the ocean, but somehow they've made it out to these islands. There are also some burrowing reptiles, like this legless skink over here. There's some burrowing snakes. There's frogs, which I've sort of alluded to. So just a number of things that you wouldn't expect to find on an oceanic island that have made it out to this archipelago. So we find that there's been a lot of dispersal in the history of these islands, and in some cases we find that there are species or sister species that are found on different islands, and so it suggests that not only has there been a history of a lot of dispersal from the continent to the islands, but that in some cases we've had dispersal between islands as well. And the amphibians demonstrate this pattern even better than just looking at the overall patterns of biodiversity. So if we look at the amphibian diversity on Bioko Island, and this is just a sampling of the species that you can find there, they represent most of the genera that you can find in adjacent Cameroon, and most of these, well, almost all of these species are species that you would find in Cameroon, just on the other side of the marine incursion. But then if we transition to Principe Island, we only have 3 species of amphibian, but they come from 3 different families of frogs. So these are presumably from 3 independent dispersal events. And then São Tomé has 5 species of amphibian, again, mostly from different families. And this thing in the middle there is a limbless amphibian called a Sicilian. And again, not something that we would think of as being a very good disperser across saltwater barriers. And then Annobón doesn't have any amphibians, so I'm not going to talk about it again today. But it is the smallest, youngest, and furthest out island from the continent, so that might be part of the reason. So again, amphibians stood out to me as a really intriguing group to look at this history of dispersal and in situ diversification. And I ended up focusing on the reed frogs, which are the only amphibian group that diversified within the archipelago. So there are 2 species endemic to São Tomé Island and 1 endemic to Principe Island. And so this seemed like a great system to look at the colonization history of amphibians to the islands, as well as dispersal between islands, and then also diversification within the archipelago. So first we'll start with the land-bric island. So there have been a few studies that have looked at patterns of genetic variation between island populations and their mainland counterparts. And for the most part, these have been good dispersing organisms, mostly birds. But there are a few studies in amphibians and reptiles. And for the most part, what people have found is that the diversity on the island is, or the divergence between the island and the continent is very recent and is sort of on par with what you might expect between 2 little hills or mountaintops just on the continent. So just regular population level divergence that is sort of commensurate with the distance and elevation between these regions. In some instances, though, we find that, or others have found that there's cryptic diversity in the Bioko lineages of what were once considered to be these more widespread species. So this study of this little forest gecko included a large genomic data set, and species delimitation methods recovered strong support for that Bioko Island population being distinct. And then when they went back and looked at the morphology, they found some morphological characteristics that supported recognizing this as a distinct species. So what this indicates is that some species seem to not really be experiencing this isolation due to the marine incursions, but there's certainly the potential for these incursions to be leading to the accumulation of endemic diversity that's recognizable on the basis of both genomic and phenotypic differences. And so I decided to focus on amphibians again. I like amphibians, in case that isn't clear by now, but also because I figured that they should be one of the more sensitive groups to these marine incursions, and so they should be the ones that are showing this pattern of species accumulation through time if the incursions are related to this. And so we can think about what these marine incursions are doing to island populations, and I've put together this series of cartoons because I think it helps demonstrate how complicated this quickly becomes. So for organisms that are poor dispersers across saltwater barriers, we expect that an incursion should lead to genetic divergence between the island population and the rest of the mainland distribution. But then when sea levels retreat, these populations can expand across the exposed shelf, and they might interbreed, and so these differences that they've accumulated in isolation might become obscured, and that would ultimately reduce divergence between island and continental populations even when that incursion comes back into place. And so this rising and retreating might sort of erase these signatures of divergence if the populations are interbreeding when the land bridge is exposed. Another option, so we know because we have endemic species on these oceanic islands that have never been connected to the continent, we know that frogs can disperse across saltwater. It doesn't seem to happen very commonly, but it does happen. And so another option for how we're getting populations of frogs on Bioko is overseas dispersal. And the demographic signatures of this should include a population bottleneck and then a demographic expansion event. And so this would differ from this pure vicariance scenario in that we would expect to see a period of exponential growth in that island population. And then, of course, we can layer onto this that there is still the potential for these discrete admixture events when the sea levels retreat and the continental shelf is exposed and those populations come back into contact. And so another option is that because Bioko is close to the continent, and again, that we know that frogs do occasionally disperse over water, that maybe these dispersal events are actually fairly common. And so that even though we tend to think of these marine incursions as being these hard barriers to dispersal in amphibians, that maybe they're not a very strong barrier to migration, and that so that there's been this history of small amounts of gene flow from the continent to the island throughout its history, even during periods of sea level rise. Of course, we would expect that there's more admixture when that sea level retreats and the land bridge is exposed, but that there's not these periods of strict vicariance. So to start disentangling what you're probably thinking is a very complicated set of demographic scenarios, we took a modeling approach. So first, we generated environmental niche models for species based on occurrence data throughout their ranges on the continent and the islands. And we then predict habitat suitability for the species through time, going back 120,000 years, to see where the distribution of suitable habitats would have been for that species under these different scenarios. And then we zoom in on the continental shelf to quantify not only when was the shelf exposed, but also what was the suitability of climate across the shelf for that given species. So here we're going to look at connectivity between Bioko and the rest of the continent in 1,000 to 4,000 year time slices for 4 species. So the 2 species on the left occur on Bioko, and the 2 species on the right are absent from Bioko. And what you can see is that there's this 50,000 or so year period where there's high connectivity across the land bridge in the 2 species that are present on Bioko, whereas for the 2 species that are absent, we have some very short episodes of intermediate connectivity, but for the most part, the connectivity is much lower. So if the marine incursions, well, and I guess then we also see that there's clearly the continental shelf was separated in recent history, but then also 70,000 years going back to 120,000 years. So we have this window of opportunity for colonization to happen, and then we would expect that the time to most recent common ancestor for these island populations dates to around this window of time, because this was the last opportunity for unobstructed gene flow. So we have some mitochondrial divergence estimates for the species that are present on the island. So here I'm showing the island populations in pink, and then the most closely related mainland populations in orange. We can see that the island populations are generally distinct from the continent, and that this divergence estimate roughly overlaps with that period of connectivity. It's at least on the same relative timescale of being a recent divergence. There are obviously caveats when using these molecular clock methods for dating recent divergence in particular. So then, when we look at the second species that's present, we have 2 distinct haplotype groups on Bioko Island, and they're again roughly within the same general time frame, so relatively recent events. And initially I was a bit puzzled by why we would have these 2 different groups. So the one option is that we had a back colonization event from Bioko to the continent. And a second possibility is that the source populations for Bioko Island came from distinct lineages on the continent. Again, when that continental shelf is exposed, it's a very large area that's potentially connected, and so the source populations could be coming from multiple places on the continent and then becoming isolated on the island. And then, so we've been expanding out to more species. So here's a species that has a pretty similar ecology to these little tree frogs. It's called a leaf-folding frog. It lays its eggs on leaves, and then it folds its leaf around the eggs with some little glue. So they're a pretty cute little egg nest to find. And you can see that, again, we have a distinct mitochondrial lineage on Bioko, and the date is roughly within the same time interval as the other species. So clearly there are some limitations to just looking at these patterns of mitochondrial relationships between these different populations and trying to differentiate between these very complicated scenarios that I was just walking you through. And so, again, I said we took a model testing framework using DD-RAD nuclear SNP data. And what we did was compare these different demographic models that can account for the demographic signatures associated with a vicariance model or a founder event model, and then layering on these different options of having low-level dispersal throughout the history of the population, and then also having these periods of admixture or discrete admixture. So here we first collect, again, genetic data for the species throughout its range on the continent and on the island, and that's to identify which continental population is most closely related to the island and is appropriate for our modeling scenarios. And then we use the two-dimensional joint site frequency spectrum to compare these different models of the joint demographic history for the species. And so in this case, we're interested in whether the population on the island arose under a scenario that's more consistent with vicariance or with a founder event. And I'll say most of this modeling work was led by my colleague, Dan Portick, who was a grad student at Berkeley when I was a postdoc there. And this work was also in collaboration with an undergraduate student who was exceptional. So thank you, Kristen. So what we ended up finding was that the best demographic model for this leaf-folding frog was the vicariance model with this late discrete admixture event. And in case you've forgotten what that looks like, here's the cartoon version. So in thinking about what the populations are doing, so we have support for a scenario of the populations being isolated by these sea level incursions, and then a period of connectivity, perhaps dating back to when the sea level most recently retreated, and then a current situation of vicariance. So because other studies had found some morphological differences between island populations and their continental counterparts, we wanted to see if there was any indication that this was true for the leaf-folding frogs. And throughout their distribution on the continent, they've been recognized for having a lot of color pattern variation. So this is one of the many things that we decided to look at. And unfortunately, we didn't find a pattern. So it looks like these color patterns occur in the same sites throughout their range, and that there's not any association with genetic divergence. And then we also took a lot of morphological measurements, and here I'm showing results for the 4 distinct genetic clusters that we found in our data set, one of them corresponding to the island, so the yellow one. And you can see that one of those clusters does have some morphological differences, but that the island one is indistinguishable from its closest mainland counterpart. So we concluded that the island population is not a distinct species. So these 3 species that we've looked at so far are pretty similar ecologically. They're similar in body size, and they have pretty similar reproductive biologies. And so we're now extending this work to a more ecologically diverse subset of the amphibian community. So we're including more families of frogs, but these are also species that differ in traits like body size and reproductive mode, because we think there's the possibility that these types of traits are influencing how they've responded to these cycles of marine incursions. And so we have data for a lot of these species, but we just haven't been able to put them through the pipeline yet. But I'm excited to see what we're going to find, especially with respect to whether the timing, again, is holding up consistently with divergence being relatively recent, and then whether or not any of these show phenotypic differences from the rest of the continental range. So now we'll switch to the oceanic islands in the archipelago, And in these cases, we know that overseas dispersal is an important mechanism for shaping patterns of diversity. So you may have been wondering, how did things get to these islands? And the prevailing hypothesis for how this has happened is assisted dispersal on vegetation rafts. And the main reason that this is the hypothesis that we're going with is that we have so many organisms that we think just wouldn't make it out there without this assistance, especially things like the shrew, again, which has to eat every couple hours, and then burrowing animals like the Sicilian and the legless lizard and the snakes. So the idea is that you have these large vegetation rafts or pieces of riverbank that are falling into these massive river drainages and being swept out to sea, and some of them make it to the islands. And there are three major rivers that reach the Gulf of Guinea that could potentially serve as sources of these types of vegetation rafts. So the first is the Congo River, which is the largest river in Africa, and it's the primary drainage for most of the Congo forest in Central Africa. So this is just a very small piece of it. It would be going all the way out to that speaker. Second, we have the Niger River, it originates in the Guinean highlands in West Africa, and it drains most of the West African forests. And then we have the Ogooué River, which is the fourth largest river in Africa by discharge, and it drains most of the rainforests of Gabon. And so in case you're having a hard time picturing what these vegetation rafts look like, this is a very gratuitous artist rendition. So that's an island in the distance. And then this piece of riverbank is basically a Noah's Ark with trees and lots of different habitats making its way out to the island. So we think it looked somewhat like this. And I will say that these types, I don't know that exactly something this dramatic, but vegetation rafts being swept out from the Congo and the Amazon, people have documented this before. And for the Amazon River, someone had a lot of time on their hands, I guess, and a very cool project idea where they were actually intercepting these rafts and documenting what they were finding on the rafts. And they did find all kinds of vertebrates and invertebrates on them. So it's certainly conceivable that this would happen. I imagine that a lot of the rafts don't make it to islands, but when you have 30 million years for something to occur, it probably does happen once in a while. So even though the mouth of the Ogooué River is the one that is the closest to the islands, it's about 250 kilometers away. The currents in the Gulf of Guinea direct freshwater plumes from the mouths of these rivers towards the islands. And so it's conceivable that rafts originating in either West Africa or even all the way out in East Africa are actually making their way towards the islands. And that fresh water coming from the mouths of the rivers also decreases the surface salinity, which can be important if you have waves that are lapping up against the vegetation rafts. An alternative hypothesis to this that my committee members were always reminding me of when I was a graduate student is that there could be island hopping, right? So we could have had some lineages that colonized Bioko, either when the sea levels were lower and it was connected, or dispersing overseas, but then that we could have this island hopping going down the island chain. And this is documented in a lot of other archipelagos and is certainly a reasonable hypothesis for this system. So we can reconstruct the colonization history of the endemic fauna based on the geographic distributions of the mainland species that are most closely related to those island endemics. And the phylogenetic studies that have included the island endemic amphibians tend to find this relationship where the most closely related mainland species are out in East Africa. And so this would invoke the Congo River as being the main source of these vegetation rafts and overseas dispersal. But a lot of these studies were limited by the availability of taxonomic sampling, and especially in West and Central Africa, there just weren't as many genetic samples available for this work. And so obviously, you can't really test this hypothesis effectively if you're not including these other regions in your sampling. And so I focused a lot of my work into fleshing out some of these sampling gaps as a PhD student. So I chose to focus on the reed frogs because, again, as I said, they had diversified within the archipelago, or presumably had diversified within the archipelago. And I was lucky that right when I was starting this project, a mitochondrial study had identified a species complex on the continent as being closely related to the island endemics. So I didn't have to reconstruct a phylogeny for the whole genus because I had already sort of zoomed in on the relevant species group to focus my mainland sampling. So the most closely related group of species on the continent are the cinnamon belly reed frogs, so Hyperoleus cinnamomyoventris, which is a mouthful. And this is a species that is found throughout Central Africa in both forested and disturbed habitats, and it's found pretty continuously throughout this range. So based on just its distribution across Africa, we can a priori rule out the Niger River as being a potential source because the species distribution doesn't overlap with that river. And then, again, we have this stepping stone hypothesis, but you may recall from the niche modeling that this is one of the species that doesn't occur on Bioko Island, so we can rule out this stepping stone colonization. And this still leaves two potential rivers as being the source for this vegetation raft and dispersal. And so what I did was take a phylogeographic approach and include sampling throughout the species range to really zoom in on which part would have been the source of the island colonizers. And so here we're just looking at a mitochondrial phylogeography of the island species in pink, and then populations on the continent in these other colors. And you can see that the populations in green are the ones that are most closely related to the island endemics, and that most of these populations are centered on the Ogooué River, so in West Central Africa. It looks like that group extends down to the mouth of the Congo River, but it certainly does not extend all the way out east. And so looking at looking at these relationships, we could invoke either the Ogooué River or the western part of the Congo River as being the source for this vegetation raft. And I think this makes a lot of intuitive sense, but up until this point, the prevailing pattern was that the relatives were coming all the way from East Africa. And so this study really demonstrated that we were going to have to take a phylogeographic approach to try to get at this question and not just base it on limited sampling and species relationships. So if we look at the pattern of divergence within the island species, what we can reconstruct in terms of the colonization history is that Cinnamomyoventris would have dispersed to São Tomé Island, that diversification in situ would result in two species, the São Tomé giant reed frog on the right and Muller's reed frog on the left, and then that dispersal proceeded from São Tomé to Principe, resulting in the third species, Hyperoleus drusei. And what is somewhat unique about this result is that it invokes dispersal to the younger island first, and then dispersal from the younger island to the older island. And this is in contrast to one of the rules of island biogeography, which is called the progression rule, where we typically see dispersal occurring from older to younger islands. And there are only a few studies that look at other organisms that would have been rafting between islands in the archipelago that invoke this pattern of younger to older colonization. But in this part of the Gulf, the currents are flowing from São Tomé to Principe. And so I think for organisms that are relying on passive dispersal, thinking about how they're dispersing and what factors are going to determine the directionality is probably more relevant than just thinking about island age. The second thing that is somewhat surprising about this pattern is that if you look at those divergent states, they're relatively recent, so occurring within the last couple million years, which is in the context of especially Principe Island, which is 30 million years old, these are pretty recent arrivals. And so one of the things that I'm hoping we'll be able to get at when we expand these methods out to a more representative set of the fauna is whether this is representative of especially the amphibians out there, or whether we sort of see this spread of dates indicating that there's been a slow accumulation of diversity over time, or maybe that there have been pulses due to certain geological or climatological conditions that would have promoted more dispersal or less dispersal through geological time. Okay, so I had this really wonderful REU student last summer, and he's starting at Michigan State University right now, so he's kind of your neighbor. His name's Kyle, and he was going to do this very cool project looking at the last amphibian endemic to the islands that we have no idea where its continental counterparts came from, or which species they are. So it's a giant tree frog. It's one of the largest species in Africa, and the hypothesis was that it is closely related to these other really large-bodied tree frogs in Africa, so Macrotus, Milsoni, and Rufus. And one of these species is distributed in West Africa, one of them is in Central Africa, and then one of them is in West Central Africa. And so what I liked about this particular group of frogs was that there was the potential to test all three rivers as a source of a dispersal event, and one of the first times that we could test this Niger River dispersal possibility. And one of these species also occurs on Bioko Island, and so we could more effectively test this island hopping hypothesis, especially because this island endemic giant tree frog is endemic to Principe Island, which is the next island in the chain. And so, unfortunately for Kyle, the main thing that we learned through his project was that the island endemic species is not closely related to either of these giant tree frog species on the continent, but we also don't know what it's closely related to. So this was just a mitochondrial study with very, very poor support for species relationships, and we basically are right where we started. But the saving grace is that, at least just from an evolutionary standpoint, what's interesting about these results is they indicate that gigantism has evolved multiple times in this group. And so Kyle was at least satisfied with that outcome and thinking about next steps for this system. So for me, what this indicated is that we needed a lot more robust genetic markers, and we are going to have to do basically a big phylogeny for this group. So here we had about half of the species sampling, but as you can see, even just looking at the ranges of these different species, there's no way that we can really zero in on which part of the continent is even relevant to be focusing on, so we're just going to have to sample all the species and do a big phylogeny. And hopefully, in the next couple of years, I'll be able to tell you where the Principe Giant Treefrog came from and when it got to Principe. So now we'll shift to diversification within the archipelago. So again, to remind you about the reed frog species, we have two that are endemic to São Tomé Island. So the giant reed frog is the species on the right, and reed frogs are not very big, so a giant reed frog is about this big. And then the species on the left is Muller's reed frog, and then the species endemic to Principe is Drew's reed frog. And the giant reed frog only occurs in primary forest habitats, whereas Muller's reed frog and Drew's reed frog are what we tend to refer to as a trash species. So they occur in really disturbed habitats, and you can find them breeding in people's backyards and in agricultural fields. They're not very picky. So now that you remember who everybody is, we're going to take a closer look at the diversification within the archipelago. So we inferred that there was in situ diversification on São Tomé Island that resulted in Muller's reed frog and the giant reed frog. And then that there was overseas dispersal that resulted in Drew's reed frog being endemic to Principe. And for the record, I don't actually think it happened this way. I just really like that little cartoon. So first we'll just look at divergence between the Drew's reed frog and Muller's reed frog, and perhaps unsurprisingly, we see that they form completely distinct genetic groups and that we don't have any indication that there's admixture between them. And so what this confirms is that although dispersal happened at one point in the history of these species, it's certainly not ongoing. And when I took a closer look at the morphology of these two frogs, so when I first started this project, they were considered one species, but clearly they're distinct in the genetic data, and I went back and found phenotypic differences between them as well. So I described the species on Principe. So you may have noticed that the giant reed frog and Muller's reed frog also differ in coloration, not just in body size. And they also differ in the types of breeding sites that they use to deposit their eggs. So the giant reed frog puts its eggs in tree hole cavities that have water in them, which is a really derived reproductive mode for frogs. And then Muller's reed frog basically will breed in any standing water that it can find. So I found them breeding in rain barrels, in a pig trough, in a ditch on the side of the road. Basically any standing water is acceptable. And in spite of these very clear phenotypic and ecological differences, my first time in the field, I found a few individuals that had intermediate coloration. And so I thought, I wonder if these guys are hybridizing. And when I looked at the genetic data, it looked like there was some introgression between the species, especially at sites where they're sympatric or in near symmetry. So I used this method called new hybrids, which assigns individuals to different classes of hybrids. So in this case, the vertical bars represent an individual, and the color is the assignment probability to different classes of hybrids. So pure pink is pure Thamensis, and pure yellow is pure Mulleri. And then these intermediate colors are either backcross hybrids to one of the two parental types or F2s. And so what we can see is that at these 3 sites in orange on the map, we have a very large proportion of individuals at these sites being classified as either F2 or backcross hybrids. And then once we had these genetic data to go back and look at the phenotypes, we could confirm that indeed those individuals are intermediate both in size and in coloration. So what's surprising about these results is that we find a really high proportion of hybrids at these sites, and they must be genetically viable because we're finding multi-generation hybrids, F2s and backcrosses. But it appears that this is a very narrow region of hybridization. And so this pattern could either reflect that there's very limited contact between the species, that it's recent contact, or that there's very strong selection against hybridization or strong selection outside of the hybrid zone. And so if we think about what's happening with the environment at these sites, the hybrid zone, at least this particular one, coincides with the transition from agricultural land to primary forest. And at these sites, when I find individuals hybridizing, they're artificial water bodies, so either tarps that have filled with water or rain barrels that farmers are using to water their crops. And so this is a very patchy environment. So we hypothesized that this is a mosaic hybrid zone and that it's potentially caused by human-induced habitat fragmentation. And so we looked at both how the genetic and phenotypic variation are tracking land use and then also some basic environmental variation in this region. So if we look at the land use in this part of the island, and here I've just numbered the sites starting from the site, the 2 sites that are in more native forests and then moving out towards the periphery of the island. And the native forest is in green, then secondary forest, and then we move into shade plantation and non-forested areas. So we just took a buffer around those localities to get an average of the land cover type surrounding the sampling locality. And then we extracted temperature and precipitation data from those same sites because as you can see just by looking at this map, there's clearly a very strong gradient in both of those variables that coincides with this hybrid zone. And so we performed a PCA to get a low-dimensional representation of this environmental variation. So this is PC1, which is 80% of the variation, and this is representing a sort of environmental gradient as we move across the hybrid zone. So, and I guess I'll explain the rationale for why we did this is that if you look at my sampling, it's not linear, but also we don't really think that the hybrid zone is linear because it's a habitat mosaic. So it doesn't make sense to use traditional Klein methods for this system. So if we line our samples, our localities up based on their environmental gradient, this is what they look like. And then when we look at our hybrid assignments along this gradient, so we can see that the pure Thamensis are over on the native forest, cool, wet side of the gradient, whereas the Mulleri are on the non-forested, warm and dry side of the gradient. And then the hybrids, for the most part, are concentrated in these intermediate parts of the environmental gradient. But you may be wondering what's happening with this site that has a lot of hybridization and yet is next to the pure Thamensis part of the gradient. And so these two sites are actually quite different. They're both at the edge of native forest and agricultural development, but the site that has pure Thamensis is a tree with a tree hole cavity with water in it, which is the preferred breeding site of Thamensis, whereas the other site is a crater lake with a wetland marsh in it, and that's much more typical breeding habitat for Hyperoleus molleri. So what we think is that to some degree, these broader environmental patterns can explain where we're finding hybridization, but that even still the exact type of breeding habitat is an important predictor. So those results were just based really on a single part of the island, and I've since been going back and getting more representative sampling. So I haven't collected genetic data from these samples yet because I haven't decided exactly whether I'm going to do more rad-seq or whole genomes. But we're slowly chipping away at getting more representative sampling. And what's really important is that we have more sampling of Thamensis and primary forest and at least 2 other contact zones between Thamensis and molleri. So we'll be able to look at parallel contact zones across the island and hopefully to get a better idea of which of these environmental variables are more important, and then also whether the patterns line up in terms of if they really do seem to coincide with agricultural development abutting primary forest habitat. So one of the things that I think is particularly exciting about this system, other than the fact that it's a hybrid zone, and I've always wanted to study a hybrid zone, so I felt really fortunate to find one, is that these regions of hybridization coincide with a really well-studied hybrid zone in Drosophila, which I didn't think that there was going to be Drosophila research that was so directly related to what I'm doing. But so this species on the left is Drosophila São Tomé, and it's endemic to São Tomé Island. It occurs at high elevations and in primary forest habitat, and it's slightly different looking from Drosophila eucuba on the right, but it has differences in physiology, and the species hybridize again at the interface of agricultural development and primary forest, which is pretty cool. And then if we layer on top of that, we have less data for some of the island bird species, but it looks like there are 1 or 2 pairs of birds on the island that also are hybridizing at the interface of primary forest and more disturbed habitats. So our goal is going to be to have parallel sampling across these different contact zones so that we can start looking at phenotypic divergence in the parental species and variation of, in fitness of the different hybrids, whether or not we see that divergence and contact is coincident across these very distinct groups of animals, or whether each species pair has its own history. So yeah, I'm very excited about what we're going to find because it's pretty rare to find suture zones, and let alone suture zones on an island. So I've been looking a little bit at some of the phenotypic differences between these 2 species, and the one that I focused on immediately was advertisement call, and that's because this is one of the most important recognition traits in most amphibians or most frogs. So in most frog species, males produce a call at breeding sites that they use to attract their mates. These calls are usually species specific, and females usually pick the calls of males of their own species over other species. The calls usually consist of 1 or multiple notes, and the main characteristics that can differ between these notes are the frequency, so a low frequency versus a high frequency note, and then also how many times they repeat these notes. So obviously, the calls are not very effective or not perfectly effective for species recognition in this system because we know we're finding hybrids. So obviously, there's some mistakes happening in species recognition. And this also does happen in frogs that are more distantly related. So this is a photo of 2 frogs from 2 completely different families that we found in a romantic situation on Principe Island. And Principe Island only has 3 species of frog on it anyway, so it's not the kind of place that you would think these mate choice mistakes are happening, but it does happen occasionally. But for the most part, the male advertisement call is considered to be a very effective pre-mating barrier. So here we're just looking at 4 species, so the 3 island endemics and their closest relative on the continent. And on the bottom we have body size, and then along the side the dominant frequency. So again, that's the pitch of the note. And we see this very strong correlation between body size and the frequency, which is a very well-established pattern in frogs. So larger individuals have lower frequency calls. And we see that regardless of the relationships among these species, it seems like body size is the most important predictor. And then if we look at the hybrid individuals, the hybrid males are intermediate in body size between the parental species, and they also produce calls that have intermediate dominant frequencies. And so just establishing this pattern was important. Getting lucky and actually finding hybrid males and recording their calls, I feel very fortunate that that actually worked out, because you don't necessarily know in the field if it's a hybrid or not. But I think this is really going to set the stage for investigating the evolution of advertisement call and also the potential for reinforcement in this system. And then now I've switched the axes, so we still have dominant frequency, but now we're looking at the average pulses per call, so the number of times that that note is repeated. And for some reason, the species on Principe Island, which is occurring without any other reed frogs around, is producing way more pulses per call on average than the rest of the males do. And so this suggests that there might be differences either in the acoustic environment on the island in terms of the other frogs that are there, or other noisy insects, or perhaps that there are differences in terms of what the females prefer on that island versus the other island. So the next step is going to be looking at female preference in these calls. So do they discriminate against hybrid male calls? Do they prefer the calls of their own species? And is there some sort of innate preference for multi-pulsed male calls, given the option to choose those calls? So today I mostly talked about, or I guess I only talked about, the work that I've been doing on this island system in Central Africa. And what I didn't get a chance to talk about, but what I'm happy to talk about tomorrow, for those of you that I'm meeting with tomorrow, is some work that led from my observations of these colorful species in the field. And so most of the reed frog species that I study are very colorful. You might have noticed that just based on the photos that I was showing. But about 25% of the species in this genus, which is about 150 species or so now, are sexually dichromatic. So males and females are different colors. And we have no idea why they're different colors or how they undergo this color change. So it's the females that change color when they become sexually mature. So that's an ongoing area of research in my group. And then more recently, this has led to a project looking at the evolution of vision in frogs, because of course, if they're very colorful and we want to test hypotheses about why they're colorful, then we need to know what the frogs are actually observing, especially in these dim light environments, which is when they're usually interacting with each other. So expect to see more on these topics in coming years. And with that, I'll just thank my collaborators, and I'm happy to take any questions. Thank you. Oh, and I will say, these are 3 other really awesome undergrads that I worked with whose work I presented today.
So if people have questions, I can run a microphone, or Dan.
I just want to ask, so do we have any idea how likely gene tree discordance is in this group of animal? Is that generally likely or not? If it's likely, then the influence on history can be completely misled, right?
Mm-hmm. So you're talking about specifically the colonization history on the islands?
Yeah.
Yeah. So yeah, the results I showed were the mitochondrial trees, but then I also had the DD-RAD data that we analyzed in a species tree framework, so using SNAP. So, yeah, I mean, in terms of the biology of the organisms, I do think that there's the potential for, you have large population sizes, and you also do have the potential for hybridization and integration even after long periods of divergence. So there's certainly the possibility to have gene tree discordance, but I don't know that it is particularly going to be problematic, especially for the island endemic species, because they're going through these sequential bottlenecks, and we see very complete lineage sorting and very low heterozygosity within each of the species. But within the frogs in general, I think, yes, there's certainly the potential.
Excellent talk. I was just wondering, is there any point in the island's history when there would have been like a ground zero in terms of seawater rise that would have eliminated the fauna? I mean, I could think about if it's 30 million years old, the mid-Miocene, there was a climatic optimum, much warmer temperatures could have...
Yeah, so presently Principe is at about 1,000 meters elevation, and São Tomé is about 2,000 meters. And when Principe was at its maximum size, it was probably even taller than it is now. But what I have thought about is volcanism and the impact that that has likely had on the fauna. So certainly in just minor and major eruptions over time, but then also thinking about how presumably there was a very dramatic event when Principe lost a lot of its surface area. I think there's some evidence that those events can just be things destabilizing slowly over time, and then a massive destruction, basically, of just losing half of the island volume and creating a tsunami. So yeah, I certainly think that extinction happened on these islands, especially over this long period of time. But that's why it'll be interesting to get dates on more of the taxa to see if everything is very recent, then I think we can make a stronger argument for extinction, whereas right now we're still just in the early stages of dating these divergences. Yeah.
Hi, thanks for a really interesting talk. I remember from like my first evolution class many years ago, a discussion of Darwin looking at bird feet as dispersers.
Yes. Yeah,
and
this is a very common question.
Yeah, I'm wondering if that's a thing for this system, and how that would change the population genetic structure.
Yeah, so no one has ever actually demonstrated that transportation of bird eggs or frog eggs on bird feet is possible, but Darwin suggested it, so it comes up a lot. So I think for most of these species, it's unlikely because the eggs are deposited on leaves over water. So maybe if the frog tadpoles somehow were on the bird's feet and got brought out there, that would be a possibility. But I think given that we see that it's not just the frogs that would have had to get out there via assisted dispersal, we have a whole fauna of things that we don't really expect to see out there. So the shrews and the caecilians and the burrowing snakes, and those are all things that are even less likely, I think, to end up on birds' feet. So I certainly think it's possible. It would be a very cool experiment for someone to do, to see if you could get frog eggs to stick to a bird's foot and survive a journey like that. And then in terms of the demography, I mean, yeah, I don't know that you would really be able to test it based on the patterns of genetic diversity. I think you'd want to demonstrate that it's even feasible, whereas at least the vegetation rafts, people have found those before, and they have found vertebrate animals on them before. So, yeah.
I know other island systems have had this issue. Have any of your islands had any accidental or intentional introductions of, say, predators?
Yes. So There are some monkeys on São Tomé Island that were introduced. I don't know, they're not very abundant and people hunt them for food. So I've only seen them once actually when I was there. And whereas on Bioko, they're naturally occurring primates and I see them all the time, even though they're also very heavily hunted. So I think they're probably not a major factor on São Tomé, but they certainly are on Bioko where they occur naturally. And then until recently, people thought that a cobra had been introduced to São Tomé to help with managing rats in the cacao plantations, which seems like a very strange thing to do, to introduce a very venomous snake. And so there are cobras on São Tomé, but recent work indicates that they're likely naturally occurring there in an endemic species. And then more recently, of course, there are rats and pigs and goats and things that are having an effect. And I will say for the Drosophila hybrid zone, the more open forest lowland species is a human commensal that was supposedly brought with humans. So that hybrid zone is supposedly recent, so within the last 500 years or so.
Has anybody ever studied the freshwater fish fauna of these islands, if there is any freshwater fish fauna?
I don't think that there are any. On Bioko, yes, okay. But I think it's quite limited. There's a crater lake on Bioko, there's a crater lake on Annobón that I'm kind of curious about whether anything is in there. And then on Principe and São Tomé, there aren't really any lakes.
What about
in the streams?
The streams, yeah. Is no known freshwater fish there? No, okay.
I don't think so. There are some estuary type species, but yeah, I don't think anything freshwater.
Is there any evidence of back colonization from Bioko, which was land bridge, back to the mainland?
I think it's a possibility in the data that I have just based on the mitochondrial patterns, but again, I don't really trust those. In the SNP data that we've looked at, in that one species, it looks like no, and in the other species, we're still looking at the data. So I certainly think it's possible. There's no reason to think that it has to be unidirectional, but yeah. Hopefully we'll know soon.
Okay, well, thanks so much, Rayna.
Thank you.
And remember, stick around for food.